genes were also expressed in the same area as the Hox genes as the somites were being formed. So I told you earlier that Notch signaling is activated in a very periodic fashion as the somites are being formed. And it turns out that the Hox genes were also expressed in the same location and at the same time that Notch genes were being activated.
Why did they have to put quail cells in a chick? Like, you could just use a plant?
So they could have, but I think the point was to show that the pre-Semitic you could trace very specific locations at specific cell types. So it's like it's a way to do a fate map, essentially. You're basically tracing where a different tissue will develop. It might be harder to do it if you just do it in another chick, because it might be hard for you to distinguish, well, did the cell that I transplanted in that region, if it migrates to another place, I wouldn't be able to know, right? Because I can't follow it. Whereas if you have something like this, where you can say, well, that's definitely a quail cell, and that's definitely derived from what I put in, then you can make more definitive conclusions. Like if they had chick cells that have two different pigments, or like, for example, in mice, if you transplant embryonic cells for a mouse that will eventually have white hair versus black hair, you can tell in the embryo where from which regions or which cells those tissues derive from. It's the same idea here with the quicken, the quail chick. Okay. So Notch genes and Hox genes were thought to be linked because they were co-expressed at the same locations and at the same time during somite formation. Okay, so Hox genes, if you alter them, you alter the axial patterning of the vertebrae, and this is called posterior prevalence. So posterior prevalence refers to a mutation that affects the anterior extent of gene expression. So for example, if you have, here we have our normal mouse, you have basically two thoracic vertebrae and then the first lumbar vertebrae here. In a mouse that has HOX-C deleted, which is expressed usually here in the first lumbar vertebrae. So if that gene is deleted, this vertebrae becomes like the one more anterior to it. So this lumbar vertebrae gets transformed into a thoracic vertebrae. So the mutation affects the anterior expression, or it extends the anterior expression of that pattern. Does that make sense? So this lumbar vertebrae is usually more posterior to the thoracic one, right? And so here where it's deleted, it basically duplicated the more anterior vertebrae and then made a new first lumbar vertebrae. Okay, so abnormal expression of Hox genes in the anterior regions lead to tissues becoming more like the posterior position tissues. Okay, so you basically extend the anterior and then you transform the vertebrae to the next vertebrae down the line. And so this is an example of a homeotic transformation, because you're changing one segment of the body to another. Okay, so somites, once they're formed, they also get further patterned. So here, again, using quail chick chimeras, it was studied, the fate of the somite cells was studied. So this is a somite. Okay, it's in the earlier embryo. It's located near the notochord. Okay, so this is a lateral somite. And it's color-coded here to show you where different portions of the somite will develop. So this orange part is going to develop into what's called the dermal myotome. Okay, and it's made up of the myotome, which will form muscle cells, and the dermatome, which will eventually form the dermis of the skin. Then there's this quadrant here in blue that becomes the sclerotome, and this is the future cartilage that migrates around the vertebrae. Okay, so there was a question as to how does the somite know that these 3/4 of the somite need to develop into dermal myotome, whereas this specific location here will develop into sclerotome. And so what has been discovered is that, yes, right, there's signaling and cell communication occurring between the different tissues that surround this somite to tell the somite or tell this region that it is in the sclerotome region and that it's supposed to become sclerotome versus the dermal myotome. Okay, so some of the genes. Okay, so if we know this because if you do transplantation experiments where you take the notochord, which is a mesodermal derived tissue, and you graft it into a different location, okay, on the developing somite, what you see is that you get the induction of just sclerotome tissue near where that notochord graft has been transplanted. Okay, so this is the control. So this is the side that has normal development. The sclerotome is usually developing very close to where the notochord is. Okay, so if you take the notochord and put another one now on the dorsal side of this embryo, the notochord is basically producing signals that induce this somite into only sclerotome. So it's getting some kind of signaling from the notochord to dictate what fate it will be. So, yeah. Okay, so this means that there's some kind of cell signaling and communication occurring between the ventral notochord and the ventral side of the somite that can be transplanted when you just replace the notochord in a different location. And notice here too, you're not getting any dermal myotome being produced on this transplanted side. So this is similar to the induction experiments that we talked about earlier, where depending on what cells are present in that specific location, you get a different, you get basically skewing or changing of the somite's fate. Okay, so last slide for today. So it turns out that the signaling pathways that we have discussed earlier also play into the patterning of the somite when it develops into the sclerotome versus the dermal myotome. Okay, so Sonic's hedgehog is a signaling pathway that comes from the ventral side of the notochord, or from the notochord, and it's thought to be a ventralizing signal. So if Sonic hedgehog signaling is activated in these cells, they become more sclerotome. BMP4 is signaling from the lateral mesoderm, and it tells this portion of the developing somite that it is a dermal myotome. And Wnt family proteins come in from the top, they think from the developing neural tube, okay, to signal to the dorsal side of this somite. So you have basically different signaling pathways feeding into the dorsal side and the ventral side of the somite that help to pattern and determine the eventual fate of the cells within this somite.